So thank you everyone for coming to hear me talk about my research. And my research was to investigate the diet of a recently reintroduced river otter population in the northern part of New Mexico on the upper Rio Grande River. First, I'll just do a little overview. First, I'll talk about the life history and the food habits of river otters in general. Then I'll briefly mention my study area, Taos County, New Mexico. I'll discuss my methods, results, and discussion, um, and then follow up with some management implications and possible future research. River otters are in the family Mustelidae, the subfamily Lutrinae, and they are one of 13 species of otter worldwide. They are the only species of otter considered least concerned by the IUCN, and they inhabit a variety of wetland habitats across the United States and Canada. As you can see from some of these pictures, they have streamlined bodies, counter shading coloration, meaning they are lighter on the bottom and darker on top. They have very powerful vibrissae or whiskers. In short, they're pretty much built for swimming and aquatic predation. They don't dig their own dens. Instead, they rely on abandoned dens of other species, typically beaver lodges, but they'll also use fallen logs or rock outcroppings as their den sites, depending on availability. The den selection tends to depend on food availability and water level at the site. The reproductive period is very, very long compared to other species, specifically mustelids. They have a large overlap between the mating period and parturition or birth. This is thought to be because of delayed implantation. This delay is linked to photoperiod or day length. Regions with very long daytimes or tend to have shorter delays or no delays at all. And typically this will be about 68 days, the whole pregnancy, 68 to 69 days. Whereas areas with very short daytimes or day periods tend to have very long delays, almost 180 days or greater between mating and birth. Females and males are typically sexually mature at 2 years. Females have been recorded to give birth at 1 year old in captivity, and males, although they are sexually mature at about 5 to 7 years of age, they're sexually mature at about 2 years of age, but they're not typically successful until about 5 to 7 years old. The litter size is usually 2 to 3 pups, but litter sizes of up to 6 pups have been recorded. Previously, not common. And the pups typically will remain with the female until the birth of a new litter. River otters are also very social, especially for mustelids. Their degree of sociality is linked not only to gender and age, but also to their reproductive capability. So single females are typically the least social, followed by females and the cubs or pups. The sibling groups are typically formed right when the female is about to give birth to a new litter, and the previous pups are, I guess, kicked out in a sense. And then there's the male bachelor groups that also form right before the reproductive period. The degree of sociality is linked to food abundance. So when there's abundant prey, they tend to be more social. When there's less prey, they tend to be less social. And social activity can typically be recorded and observed at latrine sites. Latrine sites are areas of repeated use by otters for defecating, socialization, feeding, and scent marking. They tend to be close to the water with an elevated view of the water, a possible escape route nearby, and shading or cover available. The intensity of latrine site use is linked to the availability of the shelter, food, and water. As a side note, latrine sites are not necessarily den sites, although they might have the same requirements or characteristics. An individual's use of the latrine site is also linked to their degree of sociality. Very social river otters, or when river otters are being more social, they tend to visit a smaller amount, or they tend to visit less latrine sites, but they'll do so with higher frequency. And less social, typically female river otters, will almost usually visit a greater variety of latrines, but with less frequency at each site. And latrine sites don't necessarily have to be natural sites. They can be boat launches or decks. There's no set time period for river otter visitation. Activity tends to be linked as well to human presence or human activity. So in areas with a large amount of human disturbance, river otters will tend to be more nocturnal than not. This is just a picture of a male bachelor group in Alaska. And this is one of my favorite pictures. This is actually a boat that the river otters decided to use as a latrine. As you can see, there is availability and access to water. There's an escape route. There is an elevated view of the water. And I'm assuming there's also prey items in the water that they can eat. So if it meets the requirements, it could possibly be a latrine site. River otters were historically present and abundant in the United States. They are considered very valuable pelts in the fur trade. And during colonization and exploration, it was noted that they were very abundant, and the commercial demand increased for river otter furs. This commercial demand eventually saw huge reductions in not just North American river otter populations, but otters worldwide. These are actually pictures of sea otters and some fur trappers. So we went from otters being present and abundant in every US state except for Hawaii, and across Canada, to being extirpated from 15 of the US states, as well as seeing large declines in Canadian provinces and territories. In 1976, Colorado became the first US state to begin a reintroduction or recovery program. Between 1976 and 1991, they released a total of 107 river otters in the state. And of course, other US states and provinces began reintroductions or recovery. By 2001, over 4,000 river otters had been translocated and reintroduced across the United States, virtually reestablishing the historic range of river otters in all of the Canadian provinces and territories, as well as every state in the US except for one. And this is New Mexico. They, like the United States, overall, river otters were present but also abundant in New Mexico. There are records of Spaniard colonizers describing the abundance and presence of river otters. There are also records from the native people in these regions, specifically in this region, the Taos Pueblo people, of their use and their presence that predate Spanish exploration. Unfortunately, in 1953, the last official record of a river otter was made. It was killed in a beaver trap as bycatch outside of the town of Cliff, New Mexico, on the Gila River. There were rumors between 1953 and 2008 of possible remnant river otters in the area. However, these were never verified, so they remained just rumors. In 2006, the state of New Mexico conducted a feasibility analysis identifying several regions within the state that might be good to use as reintroduction areas for river otters. Taos County was selected, and it was the first and only reintroduction site between 2008 and 2011. a total of 33 adult river otters were translocated from Washington to Taos Pueblo land in Taos County. There have been no other river otter studies besides this, and one conducted by Dr. Cox and one of his former students that currently estimate there to be between 83 and 100 river otters in this region, not just Taos County, New Mexico, but in the northern region of New Mexico, and I believe also parts of southern Colorado. There were two main speculations for the discontinuation of future reintroductions. One is the possible sensitivity of certain species, and specifically the northern leopard frog, the flathead chub, the Rio Grande cutthroat trout, and the Rio Grande chub, of which the northern leopard frog is not in the study area. But also these three species, the flathead chub, the Rio Grande cutthroat trout, and the Rio Grande chub, have not been recorded in fish surveys since at least 2007. Most likely prior to that, they have not been recorded in the area. The other speculation is the possible competition and the perception of overconsumption of game fish and trophy-sized fish by river otters, resulting in unhappy fishermen. River otters are semi-aquatic, opportunistic predators. Their diet is mostly limited by catchability, which can be described as the accessibility, availability, and or abundance of a prey item, but also they're limited by the individual's hunting ability. Diet composition across most sites in the US is primarily fish and crayfish, except for coastal areas, which tend to also include other crustaceans because of the higher diversity of these groups. Dietary shifts are pretty common within the literature. These seasonal shifts are typically due to changes in prey behavior, namely their accessibility or their abundance. For example, in colder waters, fish tend to swim slower, becoming more vulnerable and more accessible to river otter predation. And during warmer months, invertebrates, particularly crayfish, tend to be more active and more accessible to river otters, therefore more catchable. Other groups, for example, amphibians, birds, mammals, and reptiles, typically occur at very low frequencies and are often considered opportunistic predation. However, during areas where there are large numbers, there's a great abundance of these groups, they tend to be seen at very high frequencies. Some studies even have included birds or amphibians to be up to 30–40% of the diet during spawning periods or during migratory stopovers. Other groups, and I call them other prey groups because these are not necessarily unimportant or insignificant parts of the river otter diet. They're more often less represented or undetected prey groups, and these typically will include scaleless fish, insects, mussels, and other soft-bodied items. River otters in areas that don't have other predators, for example, orcas, alligators, or crocodiles in coastal regions, coastal or marine areas, are often considered the apex predator because there's nothing that will prey on them. However, we don't necessarily know if they can be considered keystone species, like sea otters, which consume sea urchins, thus affecting primary productivity. And we don't necessarily know this because each possible trophic cascade, the magnitude of otter impacts, are largely affected by the very unique ecological context of each region. So to be able to say that river otters are keystone species, we would have to know a lot more information about what they're eating and where they're eating at on the trophic level. There are three main methods to describe river otter diet, not just river otter diet, but diet in general. These are stomach content analysis, fatty acid analysis, scat analysis, and they're two less used methods, and they're typically used to complement another method. And this includes molecular scatology or genetic testing, also sometimes referred to as eDNA. and stable isotope analysis. Um, Stomach content analysis was actually one project was actually conducted in 2010-12ish by Aaron Barding, one of Dr. Lackey's students. And stomach content analysis relies on the use of digestive tracts, and we are able to investigate and identify what has been eaten day of, and this often includes undigested items, which means we can identify soft-bodied prey items. It is considered an invasive method. This relies on the digestive tract. So obviously the river otter is dead. This is typically from a trapped river otter during fur trapping season, or from road-killed river otter. The limitations include a smaller sample size. However, this can go on for over the years, so the sample size could be large eventually. But if you have one season, the sample size tends to be smaller, and we're only able to see what they're eating day of within the fur trapping season or the period where they were hit by a car. Fatty acid analysis relies on a tissue sample where the fatty acid signatures from the river otter are compared to possible prey items. This also has the capability of detecting soft-bodied items, and we can see a very long-term or what one river otter has been eating in a relatively long term. This is also considered an invasive method, either relying on a carcass or a live trapped otter. And the limitations include that it tends to be costlier. There is a lot of equipment and training required if you're trapping an otter. You have to go through IACUC and have the equipment, have the training. And also it tends to be more of a smaller sample size because you can't capture every single otter in the area. It's not feasible. Scat analysis relies on identifying hard remains within a fecal sample. We are able to provide a long-term or seasonal approach, seasonal description of the diet. It is not invasive. We don't need to go through IACUC. And however, it might not just be the most accurate description of otter diet, as it might overlook soft-bodied prey items. Molecular scatology looks at DNA in the feces. There's a greater range of detection. It's non-invasive. It tends to be higher cost, but the price of this method is dropping. There are also issues of DNA degradation. However, with DNA metabarcoding and advances in technology, we are able to overcome the issues of degradation and still have a greater range of detection, but it does still have its issues. A stable isotope analysis is most usually done with another type of method. This relies on a tissue sample and provides insight into the trophic level they're feeding at. It's also considered invasive, and it also, like any other study that relies on a trapped otter, tends to be more costly and have a smaller sample size. Going further into scat analysis, the main pros include that it's relatively easy to collect. There is a lower analytical cost. The results are also given in frequency, which is easy to calculate. And there is the availability or the ability to monitor for the long term. And it's one of the most common food habits methods. One thing I did want to add that I glossed over was this study specifically was conducted in conjunction with another study. So we were already collecting scat for the genetic portion. So it was most feasible to do a scat analysis since we already had scat collected. The cons, like I've mentioned previously, include that it might not be the most accurate description because it does ignore soft-bodied items. Easily identified items might be overestimated, even if the specific prey item is not very abundant within the diet itself. The fact that it just is really easy to identify and very characteristic it might be overestimated within the diet. Also, it's very hard to distinguish very similar prey items because we're either using just plain sight or we're using a microscope. If you don't know how to distinguish very, very similar things, there might be confusion within the genus and even with going to family. And also the size of the specific fish might create bias. Larger fish are it's possible that they might occur in multiple scat samples. And if the fish is large enough, which is not very typical for them to be consuming very large fish, larger fish might be consumed by multiple otters, resulting in one fish being present in multiple scats and thus being double or triple counted potentially. The objectives of my study were to, one, determine the annual diet of river otters in the upper Rio Grande River, and two, to describe seasonal dietary shifts of river otters on the upper Rio Grande River. And once again, the significance of this study is that no other otter study has been conducted since the 2000 feasibility analysis to understand if otters could be reintroduced to this area. Cox and Murphy have recently recently estimated the current population to be within 83 to 100 otters in northern New Mexico. And this is the first and only river otter food habit study in the state of New Mexico. All of our study, all of our latrine sites were contained within Taos County. Our northernmost site is on the Colorado, New Mexico state line. There are 16 sites on the Rio Grande, 2 on the Red River and 2 on the Rio Pueblo. Our whole study area is in the southern Rocky Mountain ecoregion. The vegetation includes shrublands, pinyon forests, and juniper grasslands. There are very drastic changes in elevation, not just getting to our sites, but between our sites. Our lowest elevation site was almost 1,800 meters above sea level, and our highest site was over 4,000 meters above sea level. Because I'll actually go back a little bit. Part of our study area includes the Wild and Scenic Rivers Recreation Area, I believe it's called. And this is an area of the river that is very fast flowing. There's whitewater rapids, and it's very hazardous to access, which limited our ability to kayak between sites, but also to hike down and walk within sites. So our sites were accessed directly by hiking to them or by watercraft kayaking directly to these sites for our southern, the southern extent of our study area, which was a lot more slower moving water and a lot safer. Once we were at our site, we collected every single scat we could find, and then when we couldn't find any more, we hiked approximately 30 meters up and down river, unless there was, we weren't able to do so because of rocks or vegetation or the river itself. All of our samples were labeled and stored individually, and then they were frozen, and until they were we were able to have them mailed to the University of Kentucky, which again, they were frozen until we could analyze them. And here are some pictures on the right-hand side of some of our latrine sites. There, like I said previously, there's lots of elevational changes there. The elevational drop for some of our sites was almost 1,000 meters from the car to the river itself. And then we had to hike along the side of the river to access them. Some scats were a lot easier to find, especially if they were on a large boulder. Other sites or other samples were a lot harder to find, especially if they were within the vegetation, like the picture on the bottom right-hand side. And this scat here, I believe, was also, we're looking at the latrine site from the scat here from across the river, just to get more of a picture of what's going on there. Once in the lab, and we're able to begin our analysis, our samples were dried for a minimum of 24 hours. Some samples did take longer. Um, for example, our crayfish samples on the bottom right-hand side, there's one very big crayfish sample. I have my pointer. This sample here would have probably taken 2 or 3 days to dry, whereas our fish sample here on the top right hand of the scat sample picture would have maybe taken 10 to 12 hours to dry completely because it's most likely all fish. We classified our samples at first as being fish only, crayfish only, or mixed samples. We then removed any and all vegetation that we could find and non-prey items such as uh, hooks, sinkers, plastic. Here we have some pictures of some plastic microbeads that we found within our study. This was a project undertaken by an undergraduate, so I will not be discussing microbeads. And we also ID'd other major group items such as birds, reptiles, or mammals at this point. Our second sorting was when we removed identifiable objects or parts such as scales, jaws, and pharyngeal teeth, and we kept those separate for identification. Before I was able to identify, I first had to create a manual because there were no regional keys for scales in this area. I was provided scale samples by the University of New Mexico's Museum of Southwestern Biology, and they removed samples for me, and they identified scales, and we had those ready as slides to compare. And the New Mexico Department of Game and Fish provided fish samples for me where I could pull my own scales and have some jaws ready for comparison. I created my short identification manual through the use of morphology, circuli, and radii. The morphology is the general overall shape. The edges, is it smooth? Is it scalloped? The radii are the lines typically emanating from the edge to the center. And the circuli are not unlike tree rings that we're able to tell if they're crossed, if they're smooth, if they're interrupted, if they're not. That gives us some clues as to who the scale might be. And then I also use the presence of teni, which are seen in this scale here, which are tooth-like projections that certain fish families have. In specific, centrarchids in my study area were the only fish family with teni, so we're able directly to identify to centrarchid just by seeing a scale. I also used online resources to compare pharyngeal teeth and jaws. We did not have samples for every fish within the fish families identified in this area. So I was only able to confidently identify a family using family level characteristics. Some of our samples did contain fish, but they were not identifiable. This usually happened when we only had vertebrae or spines, where we knew definitely that the bones belonged to a fish, but we could not find any identifiable features, and these were only recorded as fish present. Here are some examples. This is a cyprinid pharyngeal tooth. Here are some scales. The scales of cyprinids and catastomids are similar. Therefore, I relied on pharyngeal teeth to distinguish between these two families. The catastoma teeth typically occur on one plane. They're kind of comb-like in the sense where cyprinids, they have different levels, and they're wavy. They have a spatulate tip, so they're easily differentiated between these two families. Salmonids and centrarchids, however, their jaws tend to be similar. So here, whenever I would find a jaw, I would rely on the scales to differentiate between the two, because salmonid scales are very, very small. They don't have radii. They actually, if you can imagine what a fingerprint looks like, that's kind of what salmonids look like at a very, very small scale. Whereas centrarchids are, they have scalloped edges, they have radii, and most importantly, they have teni. which is a family characteristic, so we're definitely able to distinguish there. And ecocids only had one fish family, or one fish species within the family, which is the northern pike. And both their teeth and their scales are very easily identifiable, so there is no confusion between Ecocidae and any other family in the area. Prior to analysis, I first determined seasons by precipitation and temperature. I determined the dry season to be November through April. The dry season also tends to be cooler. And then the wet season was May through October. The wet season also encompasses the warmer months. And I first calculated frequency of occurrence, which frequency is often given as a percentage or a fraction, or a decimal, which is the number of times one food item occurred over all of the food samples provided. I also calculated relative frequency of occurrence, which is the number of time one food item occurs in relation to all of the other food items present, not the number of scat samples. But I'll only discuss frequency of occurrence for ease of communicating within this presentation. I then used R to analyze the seasonal differences. I used a general linear mixed effect model, GLMM, which was fitted using Laplace approximation or maximum likelihood method. I used the R package LME4 function GLMER. Our response variable was the log likelihood of prey identification. The fixed or explanatory variable was the season in which the sample was collected. So it was either wet season, so it was the wet season and whether or not it occurred in the wet season. And if it didn't occur in the wet season, then it occurred in the dry season. From there, I transformed log likelihood or logit to probability odds and odds ratio, where I'm only going to discuss probability and odds within this presentation. Further, then the p-values were interpreted to determine significance. And as for our project timeline, our project officially commenced in January 2018, where there was a preliminary survey to remove all present scat, so we would know for sure that all of the scat collected occurred during the 2018 year. Our first scat sample was collected in February, the first week of February. The genetics portion of this project was concluded in April. We collected a total of 686 samples by April 2018. And from then on, we only visited once in June, once in September, once in December. And we collected 173 samples then. By March 2019, I had concluded identification, and that's when I began my analyses. In total, 877 samples were collected between February 2nd and December 20th, 2018. 20 of our samples were excluded due to possible contamination with other samples during either collection or rinsing. There were also possible labeling errors where we couldn't understand the writing on the label, or the date was blurred, or the location was misspelled. There was also one or two that could have possibly been raccoon scat, so we excluded those two samples. In total, we analyzed 857 samples, identifying 1,695 prey items within these samples. Some of our sites were visited specifically during summer, so during June, and a couple during our December visit where no items were collected. This could either have been due to lower water levels or warmer water levels. Warmer waters tend to have less oxygen, making them less friendly for fish to survive in. But also, lower water levels are not friendly or conducive to the presence of river otters. And prior to the study, we didn't conduct any fish surveys, so we can't necessarily answer questions regarding diversity or comparing prey consumption to what's actually available, because we don't necessarily know to the full extent what is available within the river. However, previous studies have stated that have conducted scat analyses and other diet studies uh, for the consumption of river otters to almost accurately reflect what is available, meaning that river otters don't tend to target specific species of fish. However, each area has its own context, and we don't necessarily know for sure if that's what's happening here or if it's not. First, the probability of occurrence. The probability is almost reflected in our frequencies. So specifically for these, I'll just state that the differences, that our seasonal differences were significant for every fish family except for Centrarchidae. Our Centrarchid identifications were very low and they only occurred within one season. And our other prey groups, we didn't have any significance reported for mollusks or reptiles. We also had very low sample size. And for avian prey items, the model didn't converge, so we can't really speak to significance of avian prey. In our annual sample, fish and crayfish were the two main groups. Fish occurred in 61.8% of our samples. Crayfish occurred in 66.2% of our samples, so they were the most frequently consumed or identified prey item in our study. Seasonally, there is a seasonal shift in what's consumed. Fish are most frequently consumed during the dry season, which tends to be colder. So it is possible that fish are more vulnerable and more catchable during the drier season and the cooler months. Crayfish, however, are almost in 100% of our samples during the wet season. It is possible that there's huge crayfish activity, but fish are also consumed at high rates in comparison to the dry season. Yes. And our fish families, salmonids were the most frequently consumed fish prey group, occurring in 39% of the samples annually, but catastomids were consumed very close after, so they're a very close second. And the other three fish families were consumed at lower rates. Centrarchids were only consumed during the dry season, and all of our fish species were consumed at our highest frequencies during the dry season, except for cyprinids. And it is possible that cyprinids are being more consumed during the wet season, because that time period of those months tend to overlap with their spawning period. And just to compare our two main food groups, which are fish and crayfish, to our other groups, which include mussels, birds, reptiles, and mammals, there's just a huge difference between the main two groups and everything else. Overall, crayfish and fish were the most frequently consumed prey items in this area, which matches research conducted in other regions. The primary food groups, like I said, are fish during the dry season and then crayfish during the wet season. And these are significant seasonal differences in identification. The fish families all occur most frequently during the dry season, and all of our seasonal differences for fish families were significant except for centrarchids, and it's most likely due to a small number, except for cyprinids. Cyprinids had a significant difference, but they occurred most frequently during the wet season. And it's not common for crayfish to be the most frequently consumed item, either annually or seasonally. And I like to restate that crayfish in this area are not native and they're considered invasive. So it's very interesting that an invasive prey item is the most frequently consumed annually and during the wet season. Also, mussels are not detected at very high rates. So here in our study, they're detected at about 3%. And it's most likely that the mussels that I identified are Asian clam, which are also invasive. So there is room to investigate if river otters are consuming invasive mussels as well. Also, and New Mexico has about 25 species of endangered mussels and clams. So it'd be very interesting to see a different study or study type used to estimate these soft-bodied prey items. Salmonids are considered are game fish, and they were the most frequently identified prey group during the whole study. However, their scales are very easy to identify. They have a great abundance of scales when present. So when we would look at a sample, we could tell right away when it would be a salmonid sample or primarily salmonid sample, which causes concern for possible identification bias, because more abundant scales and more easy to identify items are often overestimated within scat analysis. Seasonally, they are also occurring during the highest rates in the dry season, which overlaps with the spawning period for both rainbow trout and brown trout. And cyprinids occur most frequently during the wet season, which is also overlapping with their spawning period. So it brings to question, like, are river otters targeting spawning fish? We don't necessarily know this. And if they are, is there potential for creation for a management strategy, or is there a need for this? Specifically, also, there are three families. The three families, Catastomidae, Cyprinidae, and Isocidae, they're all composed of primarily non-native fish. So an Isocidae, northern pike, is not native. Catastomidae, the white sucker, is not native to this area. And within Cyprinidae, although there are native species within Cyprinidae, common carp is not native, and it's present in this area. So altogether, these three families with majority of non-native species, their frequency is more than 2 times that of salmonid. So it is possible that non-native species are of greater importance to river otter diet within this region than native or stocked fish. So some management implications: river otters are often blamed for the lack of trophy fish and reduced fishing quality. However, most studies that report size tend to report a preference for slower moving and small fish. So we know that slower moving, not small fish, are not game fish, and they're not trophy sized game fish. Instead, they're more finger sized, if anything. So some future research includes some educational outreach. We don't necessarily, and economic studies. We don't know but specifically that I'm interested in, river otters do consume a lot of crayfish. We know there has been research before that shows that the manual removal of crayfish and other invasive does slow the density and spread. So we don't necessarily know if river otters can act as a form of biological control, but they could possibly act as a form of detecting invasions, potentially. River otters are also described as a nuisance predator who deplete game fish, and here I have some headlines specifically for this area. River otters are cute and playful, but they threaten fishing. They're causing others to worry about trout. They're decimating fish populations, and they're night poachers. But are they actually affecting game fish densities? We don't necessarily know that. And does the negative press accurately reflect public opinion or the perceptions being represented within these newspapers, news stories? And what is the economic value of a river otter? Are river otters benefiting the area by consuming invasive species? Or is there intrinsic value enough? We don't necessarily know if the perceptions the local people have and the visitors to this area have of river otters. And this educational outreach, human dimension studies, and economic studies could be beneficial in determining if there are future reintroductions to this area. And I would just want to reiterate the significance of this study. This is the first and only river otter food habit study in New Mexico. It could serve for the potential basis of future otter projects, which would include human dimension studies and other social science studies, and continued monitoring, or even the use of different methods to continue to monitor river otter diet in this area. And it could possibly inform managers whether or not to consider future reintroductions of river otters to other regions in New Mexico. That's it. I would love to thank my funding sources, the New Mexico Department of Game and Fish, University of Kentucky College of Ag, Food, and Environment, the Student Sustainability Council, my student workers, and my STEAM Academy volunteers, without whom I would probably still be in the lab to the day, analyzing and separating samples, and my field technicians that collected samples in New Mexico for me, University of New Mexico's Museum of Southwestern Biology, and New Mexico Department of Game and Fish employees who provided the samples and provided guidance for identification, as well as Amigos Bravos, Friends of the Wild Rivers, that provided our kayaking equipment while we were there. So thank you so much, and I can take any questions. If you have a question, please unmute yourself and turn your camera on.